experimental results using animals, facilitating availability, and very importantly, not just making them available, but making them accessible to researchers. We like to think of ourselves as playing a very egalitarian role of ensuring that any laboratory, big or small, can access these resources that we have in the archive. And we obtain these from donations, and these mice are deposited by researchers like you from individual laboratories to the MMRRC, which we are then charged with protecting and preserving them for research use. We also coordinate bioinformatics resources associated with these lines and add additional information to lines over time. And then also provide, as much as possible, user-friendly support, technical customer service, and so on. We are a consortium, as I mentioned, and there are 4 of us, as indicated here on the map, the red stars. So I'm the PI, the project director for the MMRRC located at UC Davis. There are 3 other MMRRCs, one at the University of Missouri, Columbia, another at the University of North Carolina, Chapel Hill, and then finally another one at the Jackson Laboratory, which, as of course all of you know, is its own entity of raising and selling wild-type mice and also some mutant mouse collections. But in addition, they have a grant, just like the rest of us have, to run an MMRRC at the Jackson Laboratory. In addition to these 4 archive and distribution repositories across the nation, there's also the Informatics Coordination Service Center, the ICSC, which is also located at UC Davis. And then that provides coordination and bioinformatics support and also the customer service activity that supports the entire MMRRC Consortium. I'll just give you an idea of our holdings. We probably are one of the largest repositories of mutant mouse lines in the world. We currently have a repository of a little over 59,000 lines. Now these represent either live mice or cryopreserved mice, cryopreserved as embryos and or sperm. But they also represent embryonic stem cell lines. And these are embryonic stem cells that have been generated by multiple programs that have then been deposited into the MMRRC, which can be readily converted into mice for distribution. And finally, and just very recently, we've also accepted a deposition of hybridoma cell lines producing mouse-specific antibodies from the NeuroMab program. Again, that was another NIH-funded program. And we then have those cell lines that can be obtained for growth in your laboratory to produce antibodies for your work. The graphs on the bottom, the one on the left shows the cumulative holdings by year by center, and the ones on the graph on the right shows the number of orders by year by center. So a very active, continually growing program, and again, really our main goal is to ensure that the resources that you've entrusted us with are made available to the research community for research purposes. As I said, we distribute these mouse models to advance biomedical research, but in addition, because these centers are located at academic centers, the MMRRCs then leverage those other resources present at those centers and make those things available to you through their MMRRC. So, for example, you want to obtain a mouse, but it may only be present as a specific gene-targeted ES cell. Well, an MMRRC can convert that ES cell, do the injections into a mouse, and then send you the mouse. That's what I mean by being able to leverage the resource services at the centers. So the MMRRCs can provide biological, genetic, and other data, research, even research services, technical assistance. Scientific collaboration is always interested in talking about that, as well as, very importantly, training and education in the use of mice in research and specific technical areas. The graph on the right shows specifically by center the types of services, value-added services that are available at each of the different MMRRCs. So as you see here by the green check marks, for example, in the first area, colony management and breeding services, most of the centers provide colony management, backcrossing, congenics, time matings, and UNC and UC Davis has just opened up its neurodegenerative service as well. So again, the MMRRC is more than mice, lots of other services and resources available to you. We have a website. We hope it's responsive and friendly, but we certainly welcome input in terms of ways we can improve it. But we have worked very hard to make the process easy for not only donating mice, but also ordering mouse lines and services. We have an API tool that allows MMRRC users to interact with the MMRRC catalog and download data for your use. And we also offer advanced and simple search capabilities, including using major mesh terms, MP terms, RRIDs. We're very taken that to heart in terms of ensuring that we have there are resources identified with specific unique identifiers to the RRID annotation. You could search through major collections such as gene traps, the Bruce Beutler cell lines, and so on, and also research applications and that. And those advanced search options are some web screenshots are shown over here on the right-hand side. You might be interested because this is the dkNET webinar. We went through and just looked to see if we could identify any mouse resources that we had related to diabetes, such as insulin signaling pathway, AMPK pathway, or PPAR regulation pathways. And we just came up with these search results for you just to give you an idea of the kinds of things you can search for, what we come up with. So in the first one, the insulin signaling pathway, identified this by searching the MP term, increased insulin sensitivity, and came up with 3 lines. For AMPK, we can search by different MP terms or mesh terms such as glucokinase activity or cyclic AMP dependent protein kinases, and several lines show up, as well as same thing for the PPAR by searching for peroxisome proliferator activated receptor. So we tried to make the website has evolved, the access to the database has evolved in such a way to make this as simple as possible, because our feeling is there's lots of websites out there, a lot of databases for you to search for, but we want to minimize how much you have to learn how to use it. And therefore, we've tried to make this as simple as possible. But even still, if you still can't find what you need or not sure how to use it, very simple, just call or contact us at service@mmrrc.org. It goes to ICSC here at Davis, and we'll respond to you usually immediately, or certainly in less than 1 business day to help you out in finding any strains that you might have interest in. We also did a download of just the most popular MMRRC lines related to diabetes, and we prioritized the popularity based on the times they've been ordered. So you can see here in the columns on the left is the strain ID, the strain name here in the middle, and the number of times it was ordered here on the right. So again, there's a number of things, I think, a number of mice lines here in the MMRRC that would be of interest to you in diabetes-related research. Now, going a little bit more down into how you request MMRRC, either mouse strains or ESL lines, or even the hybridoma cell lines. This is the screenshot of the website for where you place an order. So you can use the catalog search, and you can select each line that you wish to request by clicking on that shopping cart icon on the left-hand side. Again, you'll notice it's very similar to Amazon or other type of web services provide shopping opportunities, again, to try to make it user-friendly. A star icon, a yellow star icon with a plus indicates the line is not yet available for distribution. So you might ask them, well, why is it appearing? Well, it may not be yet available for distribution because we just received it from the donor. It's in the process of that mouse line being shipped to the one of the centers that it was assigned to. It's going through some quality control steps before we then make it available. But as soon as we know it's accepted, then we want to put it on the catalog so you're aware it's there, and you can register interest. So if you click on that, then you will receive an automated email when that particular resource is available. You get an email saying it's available and it's ready to order. If it's available and you clicked on the shopping cart and you're ready, you can then proceed to the checkout by clicking on the checkout. The green checkout item is indicated here. And then you go through this very simple checkout process. So step one is regarding products. You select the quantities of the items you would like for each strain. Very simple. And you proceed to the next step by clicking on the next button. And then here it's regard to contacts. So you select the appropriate contacts for the order. That would be the recipient, principal investigator. Let's see, if you're a technician ordering it for your PI or a student order for your PI, then you put your PI's name in there, the billing contact and the shipping contact. The veterinary contact is optional, but it's particularly helpful when requesting live mice for shipment, because we need to ensure, and the veterinarian there at your site will want to ensure the pathogen status of the mice. And the mice are shipping from SPF facilities here. And then, of course, you can have multiple contacts under each contact type. So once that's done, you move on then to the payment, and you indicate your intended payment method. It could either be a purchase order, a check, wire transfer, or we also accept credit cards. If you're NIH funded, we really do request that you put in your grant number, and that helps us to be able to report back to NIH the utilization of the MMRRC by NIH funded researchers. And to the extent to which we can identify the specific institute, very, very helpful, because that way the NIH can get a better understanding of how valuable this resource is for them and for you. And then vendor information for the MMRRC Center. Fulfilling your order is listed at the bottom of the page. Several of the MMRRCs are what are known as approved vendors, which means that there has an agreement between the MMRRC and your institution to be able to accept live mice directly into your facility without going through quarantine. And that's a really big time saver, so that you have access to your mice very, very quickly for your research activity. Each of the MMRRCs works out a mechanism by which your institution can recognize this as a approved vendor. They, again, each institution may have specific eligibility requirements for that, primarily dependent on the pathogen status of the facility from which the mice are coming from at our MMRRCs. This is typical for commercial entities. We're not a commercial entity, we're an academic resource. But we do try to operate at an enterprise level, and using best practices to try to get mice to you as rapidly as possible. So one thing I would suggest is that if in the process of requesting a strain, you've determined or find out that your institution is not an approved vendor, then you can ask your campus veterinarian to contact us, and we can again, whatever center it is, whichever MMRRC center it is, and ask them to contact us to make us an approved vendor. Again, that will facilitate you getting access to mice as rapidly as possible. And we'll do everything we can to make that happen. The fourth step is a lot of the legal stuff. So you need to complete a legal agreement for all materials. There's a specific agreement will be sent to you once your order has been submitted, and this is generally for those lines that we distribute that may have licenses associated with, so on. You want to indicate the tax status of the institution. It's generally not for profit. Thirdly, you want to indicate that you are authorized to purchase for your institution, that you accept distribution terms of use, which are very, very straightforward. Provide the submitter contact information. The customer's internal reference number is optional, again, if you want to, and indicate whether your facility waives quarantine for MMRRC animals and whether a health status report is required. So of course, you can only answer those questions if you are authorized to purchase. And in most cases, for me too, if I was to purchase, I'd have to go through my campus veterinarian's office to purchase, so you'll want to have them fill this out for you. And then you need to provide the courier or courier account, and then the shipping instructions and the special requests. The step 5 is you just need to review your order and make sure everything is entered correctly, and then submit it by clicking on the submit button at the bottom of the page. And then the step 6 is the order confirmation. The confirmation email will be emailed to you with the order number. The MMRRC Center that fulfills your order will then contact you with further details. You can check your order status in the My Order section of the MMRRC website. And of course, if you have any questions through any of this process, anything isn't working right or it's confusing, just call customer service at the 800 toll-free line or the local number, the non-toll-free line for international calls, or again, you can email us at service@mmrrc.org. Now, that's for requesting lines. But the other side of the coin is you are a research laboratory and you have a line that you'd like to deposit into the MMRRC. Why would you want to do that? Well, let's say you're an NIH-funded investigator. Certainly, part of your NIH funding, you have an obligation to share not only data but also products that are developed through your NIH grant, including animal models. And so the MMRRC provides this as a way to fulfill that sharing responsibility, number one. Another reason might be is that you're getting a lot of requests for your line, but as a busy researcher, you don't want to be having to spend time on fulfilling those requests and going through the process to dealing with those. And that's another reason why the MMRRC is there to help get your line out there for others to use, especially after it's published. As you know, you're going to get requests from investigators who want to have access. Thirdly, maintaining mice on the shelf at your organization is probably it's the expense of doing that for your own research is probably sufficient. But to maintain mice for others who want access to them can be quite expensive. And so getting those off of your dime and putting it on us, we can maintain those mice and make those available for requesting investigators so that you can reduce your colonies. And then let's say you're done with your research on this particular mouse and moving on, but you don't want to lose it. You spent all the time making it. Well, again, sending that to the MMRRC ensures it's protected in perpetuity, certainly as a cryopreserved archive, and also for continued sharing with the research community. So a lot of reasons why you want to submit your line to the MMRRC. And again, just like we've done for the ordering process, we've tried to make the deposition process very, very fast and easy. There are 3 different types of submission processes. We call it type 1, and that is just you as an individual researcher comes to us and wants to deposit your line. There's a type 2, which you wouldn't be familiar with, and that is an opportunity we give to the NIH program officers to allow them to select 3 or 4 lines a year that they may want to ensure get deposited into the MMRRC. And at that then is a faster shot in order to get into the MMRRC. That's something that you can maybe talk to your program officer about. I will tell you that most program officers who have taken advantage of that type 2 submission process have done that for internal NIH laboratories that are working on mice to get those mice in. And then there's a type 3. Now, the type 3 is what we call a contract submission. And now this goes back to what the cost would be for depositing your line. There's no cost to you other than your shipping costs of the mouse to the MMRRC Center. Other than that, all the other costs are borne by the NIH grant supporting the MMRRC. Now, because we have a lot of submissions, we go through a monthly process by which we review and analyze those applications for submission. And not all of those lines are accepted. That's number one. Number two, sometimes some investigators have very, very large collections of mice or of cells, which as a group are far more valuable to be placed into the MMRRC for distribution as opposed to breaking it up, or a large collection that they don't want to maintain anymore at their location. They'd like the MMRRC to take it over. And in most cases, those are not accepted as a type 1, but they are accepted as a type 3. And what that means is, this contract submission, is that if they are deemed scientifically of value and meritorious, then the MMRRC can enter into a negotiation with the investigator to provide a little bit of help and support for getting those mice in the MMRRC. It could be monetary. You pay for some of the help in getting them deposited, getting them cryopreserved, and so on. It could be an in-kind contribution. Let's say a batch of cell lines, and you provide a tank for maintaining those in the archive, something like that. So it's very, very open and flexible. Again, with the goal of getting highly meritorious, scientifically valuable mouse resources into the MMRRC archive and making them available for researchers. Okay, so let me go through this little 60-second tour, which is a video of the process, which shows you how to donate a line. So this shows you where you click on the submissions, and then you start your submission. You go to the welcome page and go to start. And here, then, you tell us about who you are, who the strain developer, very simple, who the strain developer is, and you finish this page and hit next. You go on to the repository. We ask if your strain has been distributed by another repository because we prefer only to take strains that are unique. Click next. And it is the development of the strain described in a publication. Publications aren't required, but they do help in the donation process. And then we want you to describe the phenotype of the strain. That's very, very important, especially if the strain is not published. Also, then we need some allele information. What do you call your mouse? Again, very simple information requested there. And then finally, the legal stuff in terms of your agreement to donate and accepting the donor MTA conditions. And if you have any submission questions, you can contact us again and ask for help right there from the website. As I mentioned over here, some of the benefits of strain submission, they're all listed here over on the right. Also, at the very last point, as a donor, you oftentimes are able to receive a one-time, no cost to you ordering of your own mice, if it's available live, or an aliquot of cryopreserved germplasm, such as sperm, for each strain you donate to the MMRRC. And again, the only charge to you would be for shipping. So that's another nice feature that we provide. Okay. We try to engage in a lot of outreach and awareness so that you know about us. And again, this helps you as a donor so that you know that we're trying to get people to know about the MMRRC and then can find your mouse, but also for those people who are looking for mice so that you don't have to make it internally. You know then that the MMRRC is there available, and we very well, with 60,000 alleles, we might have the mouse you're interested in. And also the potential for identifying research opportunities. So we have this booth, which travels around the country every year. And here I've listed about eight or nine of the research conferences that our booth will be presented at in 2020. So we go from endo to experimental biology, the TAGC meeting in DC, DDW, ADA, developmental biology, Society for Neuroscience, American Society of Human Genetics, and finishing up the year in Atlanta at Obesity Week. So we hopefully will see you there and welcome you to stop by and to see us. And again, more outreach. We have a lot of information partnerships have indicated here. We work with, as you know, dkNET and also with the mouse metabolic phenotyping centers. We work very closely with the International Mouse Phenotyping Consortium. For those of you who are not familiar with that, the IMPC is an international group that seeks to knock out and phenotype every human orthologous gene in the mouse genome and making the mice and data available to the research community. There are 3 NIH projects participating in the International Mouse Phenotyping Consortium. And those 3 projects are located at one at Davis, one at Baylor College of Medicine, and the other at Jackson Laboratory. All the mice that are produced by those 3 programs known as COMP, all those mice are deposited in the MMRRC and made available to you. We also work with the Illuminating the Druggable Genome project. Several of the projects there are also making mice, and those will be deposited. Many of those are deposited in the MMRRC. So this is just some examples of the different information partnerships that we work with, and again, to try to get mice and information into the MMRRC. We have social media as well through Facebook, Twitter, and others. We also regularly send out email blasts. Hopefully you've received some of these in a targeted way, and an example of that is shown on the right, where we introduce ourselves to the MMRRC. We also talk about the benefits of strain submission, and oftentimes you'll receive this if we see a publication of a mouse that has been generated, and it's a new description of a mouse, a newly generated mouse, and so you'll often receive this as a request for you to consider depositing them in the MMRRC. I'll mention that also as a reason why we don't require a publication of your mouse in order to deposit it in the MMRRC. And the reason is because if you submit an application to deposit, it's found to be not available elsewhere, it's unique, scientific, meritorious, and so on, then, and we accept it, then it gives you an opportunity to actually put into your publication that this mouse is available from the MMRRC. It'll already have a catalog, RRID number, so it makes it very, very straightforward. We provide a statement that you can copy and paste into your paper to talk about where to find your mouse. And then we also have announcements that we send out regularly. Here's one for March, is National Kidney Month, and in that announcement we can indicate some mouse strains that might be of use and interest for kidney disease research. So that's it. I want to thank you very much for listening to this brief overview of the MMRRC. Again, if you are interested in donating or requesting a mouse strain, go to our website at www.mmrrc.org. Follow the links, but if you want more information, you can write to customer service at service@mmrrc.org or call the 1-800 number that's showing on your screen. So with that, I'll stop and then turn it back to Kawea and see if there's any questions from anyone.
Yeah, now you can, for the attendee, you can unmute yourself if you have any question. So the unmute button is on the lower left side of your meeting controls.
Hi, Kent, can I ask a question? Can you hear me?
Yes, I can hear you.
Okay, so when someone wants to deposit a strain in the MMRRC, what is the typical reason why it would be declined as a submission? Yeah,
good question. The strain, it may already be available at another repository, and therefore we don't want to, again, we need to be good, we want to be good stewards of the federal funding we receive from the NIH, and so if the strain is already available elsewhere, then it will likely be declined. Another reason is that while we are happy that the depositor is trying to deposit it, they may not be the originator of the mouse and may not have the right to do that. And so we oftentimes say, this doesn't look to be your mouse. You may have gotten it from somebody and used it. But so maybe we find it, we'd be happy to take it, but you'd have to get the originator of the mouse to agree to donate it or to give the authority to donate it. That's another reason. A third reason is that it may be a mouse line that is a very slight variation of what is already there. We have 5 basic criteria that we look at for a mouse line to be accepted. 1, it has to be genetically unique. Number 2, it should be phenotypically identifiable. Number 3, it cannot be available from another repository or located elsewhere for ordering. Number 4, hopefully I can remember all these. Number 4, oh yes, you agree to the terms for deposition and making it freely available. We do have now opportunities for licensing if that's what you want to do, and oftentimes we distribute it upon your permission. We distribute to commercial entities, and therefore we give you the right then to license that access to your mouse to a commercial entity if you want. So those are the, I think I said 5, but I've only mentioned 4, and I can't seem to remember the 5th one, but it's relatively minor. But so those are the reasons that we, the things that we look at to determine whether mouse line can get into the MMRRC. I tell you also that sometimes these are mice that are obtained, let's say a mouse is obtained from another source, let's say a commercial source, and it's combined with another mouse. Now, let's say we have a double knockout. Well, that would prevent us from accepting that line because the commercial source would, would say that we do not have the right to accept that and distribute that. So that would be another reason. But I would say, we accept far more strains than we decline. And then we do try to work with the investigator in those cases where it's declined, and if there's any way we try to overcome some of those reasons, we'll help you out.
And I wondered if you could walk me through the sort of quality control, like the genetic quality control you
Sure.
do on the strains.
Yeah, so we do request from every investigator to provide us a protocol to allow us to do genotyping on a mouse to ensure it is what they say it is. We then, for every line that comes in, we then use that protocol to verify that what we're receiving is what has been deposited. But we also, in addition, we do some bioinformatics analysis to ensure that the protocol itself is actually identifying the mutation that we're being provided. So we do a lot of that back-end information as well. So we do that for every line. We then will make the line available usually as a live colony, at least for the time it takes us to build up a cryopreserved archive. And during that time that we're making we're cryopreserving it, be it as sperm or embryos, again, depending on the mutation, we will then recover whatever we've cryopreserved to ensure that it can be recovered. And that makes us feel confident that if we distribute to you, if you decided to accept a cryopreserved sample of embryo or sperm, that you will be able to recover it as well. We also ask for the pathogen report from your facility before we receive it. It doesn't mean that if you have a pathogen, it's not to exclude your ability to send us mice, but it allows us to know how best to handle the mouse upon arrival. We then also either rederive it or take it through processes in order to ensure that it is SPF. So we have a list of accepted pathogens that we maintain in our facility, just like all the other MMRRCs, to ensure it's also free and clear of adventitious pathogens. We've also instituted a couple of new things over the last year or so. One is doing a mini MUGA, which looks across the genome of SNPs to identify very specifically genetic background, and we provide that as a report on every line that's deposited to us. And in addition, it also looks for specific markers that may have been used during the process of developing the mouse. So for example, let's say you got a mouse that was made from a comp ESL line, and you generated that mouse, you deposit now the mouse line back to the MMRC, and you can do that too. Well, we're going to want to be sure that there's no CRE, let's say, that's still present, or there's no additional mutations that might be present, and so that MUGA will help identify that. And then also we have begun to provide a microbiome analysis for investigators. Of course, we all know the importance of now microbiome, understanding gut microbiome at the very least in research. So what we do is we allow the researchers to send, for your donated lines, send us samples of feces that we can then do microbiome analysis. That then is part of the data that goes along with your line that enters into the archive. That way, when someone orders that mouse and gets it back to their facility, we then offer to the person requesting the mouse, we can do a microbiome analysis of your mice in your facility. Now, why is that important? Well, the mouse that was donated to us had a specific microbiome, which was associated with the phenotype that was described maybe in the paper describing that mouse. Well, when an investigator who orders it gets it back to their facility, they may want to see is if the microbiome is significantly different or what the microbiome profile is, especially if the research results they're getting are in some way different than what was seen in the original paper. So that's another feature that we offer. So those are some of the, the general things that we do to ensure quality control of lines coming into the MMRC.
So if you have any more questions, please feel free to send us emails after the presentation, and I will forward those emails to Dr. Lloyd, or you can contact MMRRC directly. Thanks for Dr. Lloyd for the great presentation, and thanks for everybody joining today's webinar. A recorded webinar should be available next week on the dkNET YouTube channel. We'd like to encourage you to share today's webinar with your colleagues and students. Our next webinar will be on February 28th. And then the topic of our next webinar is Type 2 Diabetes Knowledge Portal. To receive upcoming webinar information, please subscribe to our newsletter or check announcement at dkNET.org. Thank you again, and then have a great weekend. Happy Valentine's Day.